On this week's GARNet Research Roundup, I'm really delighted to be joined by Devin O'Connor, who's working at the Sainsbury Lab in Cambridge. And we're talking about a new paper just published in ELife, the title of which is Cross-Species Functional Diversity Within the PIN Auxin Efflux Protein Family. So thanks very much, Devin, for chatting today. And it'd be great if you could give us an overview of what you found out within this interesting paper.
Sure, my pleasure. I would say that the first thing that we were really trying to address was, are there functional differences between these two clades of PIN proteins that we identified in angiosperms? And just a little bit of backstory, we found this clade that's placed sister to the PIN1 clade, which has been lost in Arabidopsis, but seems to be conserved in literally every other angiosperm I've looked at. Mm-hmm. And so we're using Brachypodium, which is a very tractable system, to sort of address this lost clade of PIN proteins and what is their functions. And then we bring that back into Arabidopsis to test functionality and, look at what elements of these PIN functional differences are conserved. So the first part of the paper is really concerned with looking at the functionality of the two clades in Brachypodium. And so CRISPR, the miracle worker that it is,
yes, indeed.
Was able to, this was literally first try stuff. We were able to CRISPR single mutations in all three pins,
okay.
And that is the genesis of a lot of the phenotypic analysis. The big first tell from the figure 1 here is that essentially when you mutate these three Brachypodium pins, that is sister pin 1 and pin 1A and pin 1B, mm-hmm. Really it's only the sister protein that has a dramatic effect on organ initiation. Okay. And this is somewhat surprising because in Arabidopsis, of course, we all know that when you mutate pin 1, you end up with a pin, and that's the way these pin proteins were named, and it's a barren meristem. So you'll see here in figure 1 that when you mutate sister pin 1, we get a very similar defect. We look a lot in the inflorescence, and these are spikelet meristems. You can see nice barren spikelet meristems and a real brutal effect on organ initiation with cystic. But what is surprising is that actually this doesn't occur when you mutate pin 1A or pin 1B. So you can see that we have pin 1A and pin 1B single mutants. They have really well-typed inflorescences, and then even when you make pin 1A and pin 1B double, so this is a loss of function of the entire pin 1 clade, organ initiation is pretty fine, and of course we've quantified this with terminal spikelets and total spikelet number, etc.
So you found out, therefore, that this sister of pin 1 seems to be the most important pin 1 within the Brachypodium, and then you move on through the paper. You look to see, if this is equivalent in Arabidopsis, to see if this sister of pin 1 has any effect in Arabidopsis. Can you go through those experiments that you did?
Sure. So then we basically said, okay, well, if sister has an organ initiation defect in Brachypodium, what is the function when we try to look at what it does in Arabidopsis? And remarkably, it behaves like you would expect, and that is when we put sister pin 1 under the pin 1 promoter and we image it in inflorescence meristems, it makes these convergent polarization patterns, which is presumably concentrating auxin necessary for organ initiation. So even though these plants have been separated for 200 million odd years, mm-hmm. Sister retains this convergence point function, whereas pin 1B doesn't. And one of the interesting findings is that the reason that pin 1B is kind of unable to make these convergent polarization patterns is because it really doesn't accumulate well in the epidermis at all, whereas sister is able to accumulate quite well. Pin 1B just doesn't. So it took a long time to figure this out, actually. Mm-hmm. A lot of events, a lot of screening plants, and a lot of sort of imaging really big sections of plant material to see why we couldn't see convergent pin 1B. Yeah, to move on through the paper, it's probably not too much a surprise that because sister can make the convergence point and pin 1B can't, that has a functional consequence. So when we try to complement the null pin 1 allele, sister is able to do a reasonably good job and makes organs, whereas pin 1B does not, and essentially these pin 1b expressing pin meristems look indistinguishable from the null allele. So what I think this is exciting, this functional assay, because essentially we have 2 pin clades that one can perform these convergence points and mediate organ initiation, and one cannot. And this, not to get too ahead of myself, but this assay is going to be really important for our future work as a mechanism for trying to distinguish what are the protein qualities that are necessary for organ initiation and which are not.
This must have been quite relatively surprising a finding, I guess, since the pin 1, the brachipodium pin 1a and the pin 1b, they are most similar, as their name suggests, to pin 1. So there must be some aspect of the sister of pin 1, some aspect of the sequence which is allowing it to rescue in Arabidopsis, but not the others. And that's something maybe that hasn't come out of this paper yet, and maybe you're looking at in the future? Is that...
Exactly, yeah. We really believe that there are protein-intrinsic qualities that make these pins different, and it's in the proteins themselves. And of course, there's a lot known about various post-translational modifications of pin proteins. All I can say is there's no smoking gun that says, like, this domain is different in sister versus pin 1 or even pin 1a. Pin 1a and pin 1b, but we are starting to chop them, swap, and move bits back and forth. And the nice thing is then we can move back into this functional assay and try to determine which points are necessary. But as an aside, it is a little bit confusing, and I'll grant that it's confusing, that we have a pin 1 clade member, pin 1b, from brachipodium that's insufficient, and a pin 1 clade member from Arabidopsis that seems sufficient. They are in the same clade. I believe that's real and that's true. Yep. But when you stack up, and this is somewhere deep in the supplementary, I believe, when you stack up all of these Brassicaceae pin 1s and you compare them to pin 1 members that are outside the Brassicaceae pin 1s look different. Okay. And what that suggests to me is that there's probably some form of neo-functionalization, that is an acquired ability to mediate organ initiation in the Brassicaceae that's not present, at least in brachipodium, is what our data would suggest. So, what it suggests is that there seems to be a fluidity in these pin proteins with respect to their functional abilities, and I think a lot of that has to do with these post-translational modifications.
You just mentioned that when you express the pin 1s in the epidermal layer, that is sufficient to rescue the Arabidopsis phenotype. So is it only in the epidermal layers? Have you tried to express in kind of the L2 or the L3 layers within that meristem, and do you get to see something similar, or is it really very specific to the epidermis?
So we haven't done quite that experience, but I can point you to a paper from Chris Kuhlmeyer's group where they start to mess with the abundance of pin 1 in the epidermis versus pin 1 subepidermally, and that data really does point to the epidermis being really, really important for function. And of course, we tried to address that with this ML1 expression system by taking the 2 pins, expressing them under ML1, and trying to see if we really force pin 1b into the ML1, into the L1 layer, will it form convergence points? And the answer is no. It still can't form convergence points. Was again one of these pieces of data that really suggests the protein intrinsic quality. That expression doesn't matter. But no, we haven't really messed with abundances in the epidermis versus internally. But what I can say is that we've expressed these 3 different pins in all kinds of different ways. Okay. And what I can say is that the transcriptional element, while it has a role, the post-transcriptional control of these pins is the major driver of their tissue-level abundances. Okay. So we can express with all kinds of different promoters, and in certain tissues, certain pins just will not accumulate no matter how much we express them. And I think that's reflected in this ML1 data. So, back up and figure whatever 1 it is, you'll see that we are sort of trying to suggest that the pin 1 expression domain is much broader than the pin 1 protein accumulation domain, and that we kind of really need to be clear about separating these things. So
a final question for you. So if there's something to do with the extrinsic quality of these proteins. Could you do the opposite experiment? So could you express Arabidopsis pin1 in the sister of pin1 Brachydipodium mutant, and will that do rescue, or are you doing that at the moment, not to get ahead of the next page?
We've tried that so many times,
right.
and it's remarkable that it's that trying to express Arabidopsis pin1 and Brachydipodium has had more callus thrown at it than any other thing I've ever done. And this is through a collaboration with John Vogel, who has just had a major transformation pipeline for years and is very good at doing this transformation. And we just did not recover that many events. Right. So we did recover a single event. And when you image this, the pin never makes it to the membrane.
Okay.
So I don't know what is going on there. There could be any number of different things. There are codon usage issues that could be a problem. But really, it's a bit confusing because people have taken Arabidopsis pin1 and put it in tomato, for example, or in Medicago, for example. And those, not only do they show good polarization, but they do have functional they're functional to complement mutants, etc. So, why Arabidopsis pin1 just doesn't seem to work in Brachydipodium, I have no idea, but we're going to keep trying.
It's almost as if we don't have a full understanding of all the systems or something. Really.
Oh, yes, yeah. No, I think all bets are off, quite frankly.
Right. Okay, so thanks very much, Devin. We've had that's really nice discussion both of this paper and of your thoughts in this field, and where your research is going. So it's been great to chat, and thanks very much for giving us the opportunity to do that.
No, it was my pleasure. It was lots of fun.